Ani. My name is Dr. Christina Semenyuk. I am the director of FishCAST. I'm an associate professor at the University of Windsor. And it is my absolute pleasure to introduce you today to our speaker, Dr. Daniel Heath, an integrated biology professor at the University of Windsor's Great Lakes Institute for Environmental Research. Daniel has extensive experience in large collaborative projects in fish conservation, aquaculture, and ecology, using both molecular and ecological genetics and genomics. Dr. Heath's genomic research in fisheries, aquaculture, species at risk, aquatic invasive species has impacted policy and research, not just across Canada, but globally as well. So not only are we pleased to boast Daniel as one of our core FishCAST academic members, but Daniel's also very busy as director himself of his own Genome Canada-funded collaborative research program, which is on the Genomic Network for Fish Identification, Stress, and Health, better known as GenFish. And this project actually involves Canadian researchers from coast to coast, tasked with developing user-friendly toolkits to improve freshwater fish monitoring and to assess freshwater fish health. So absolutely, we have no one more perfect to talk to us today about genomics in fisheries and conservation, both the power and its limitations. Before Daniel begins, I do want to say that despite him being so incredibly busy in his research and being a mentor to so many, myself included, Daniel's able to find balance as a gentleman farmer, raising shiitake mushrooms on his farm in the eastern townships of Quebec. And with that, I'm going to pass it over to you, Daniel, and welcome everybody.
Thanks, Tina. The only time you're ever that nice to me is when you're introducing me for a talk. And I may be a farmer,
One-time deal.
No gentleman. Okay, I'm going to share my slides. Good.
Looks great. Perfect.
Excellent. Okay, so FishCAST asked me to talk about genomics in fisheries and conservation. And I thought long and hard about this. And the presentation I'm going to give you is more describing what genomics is and the power of genomics, and try to give you some idea of limitations. If you've got a lot of genomics background or interest, then of course there's a lot more depth you can get to. But the intent of my talk is to be more of a broad overview. The entire talk is going to be couched in the framework of GenFish. As Christina said, it's the Genomic Network for Fish Identification, Stress, and Health. It's a Genome Canada and Genome Ontario, Genome Quebec funded project. And it's going to creep in the slides or in my talk off and on. And I'm going to be using the GenFish slide background to keep reminding you that, in fact, a lot of the ideas I'm presenting to you actually formed the genesis of GenFish. And as Tina said much more eloquently, I do want to acknowledge that I am on the traditional unceded territory of the Three Fires Confederacy of First Nations. And it's important to realize that this has to be more than a slide at the beginning of your talk, but it has to be a way of rethinking not only how I think about science, but how I act within science. And so it's a daily process, not at the beginning of the talk process. Limitations of actually giving a talk is, here's the slide. Okay, so a little bit of background. Freshwater fish management and conservation is a real challenge, and there are a number of reasons for that. I'm going to be talking about a Canada-centric view of freshwater fish conservation and management, but fundamentally freshwater fish and their culture have incredible economic, social, and cultural value, not even talking about the ecological value of these communities. So the stakes are high. When you compound that with a country like Canada that has over 2 million lakes and uncounted numbers of rivers and streams, we recognize this is a management or conservation nightmare. But it gets worse, because freshwater ecosystems are experiencing what's referred to as the invisible collapse. It's not invisible at all, but what it is, it's the collapse of biodiversity at an unprecedented rate in freshwater systems, even relative to other ecosystems. And the reason why it's invisible is people aren't paying attention to it. And part of the reason is just the logistic barriers that I described. The fact that we're not talking about one big ecosystem like a marine ecosystem or a coastal ecosystem, but multiple millions of individual ecosystems that are connected or not connected, which makes the management and conservation difficult. It's even hard to know what's going on, much less manage it or try to stop it. This leads initially many years ago to my recognition that managers and conservationists need high quality data that allows them to make informed decisions and take informed actions. But the data has to be accurate, and it can't be simply focused on the lakes or streams in their backyard. We have to be looking at larger processes and larger patterns. What type of questions do we critically need to ask or answer using some kind of new technology? Questions such as, are there undocumented species at risk? So the graph that you see on the right here is the number of fish species at risk, and you can see us in Windsor here are in the black zone, not good. But in fact, the fragmentary nature of the map that you see probably has more to do with just the difficulties of sampling all those places and finding the species at risk. So a fundamental question, are there species at risk that we just don't know about? And the flip side of that is, what about invasive species, aquatic invasive species? Are they in lakes that we don't know about? What about exploited populations? So these are ones either commercial harvesting or recreational harvesting or other impacted by human activity populations. Are they declining? There are key stocks that fisheries managers are really interested in, and they follow it really closely. And there's big panic bells going off when the whitefish stocks decline in Lake Huron. But there are lots of other exploited populations that are not getting that level simply because of resource limitations. If a population is undergoing decline, and I don't care if it's exploited or ecologically important or even a dumb fish species that nobody cares about, if it's declining, what are the probable causes of that? If we create the technology to allow us to identify declining populations, can we also use technology to find out why are they declining? It doesn't do anybody any good to say, yep, those fish are getting tanked, unless you say, okay, what is causing it? And then you can start making positive and impactful decisions to reverse that decline, or at least stop it. And then you take the actions and say, okay, I just did something, I'm going to go back and have a scotch and relax. The question is, did your actions actually have the intended outcome? So how do you monitor the outcome of remediation efforts, whether it be reintroduction or supplementation or stress relief actions of any kind? So the key data gaps for management and conservation of freshwater fishes in Canada, in my opinion, came down to three fundamental problems. We've got all of these lakes and rivers. We need some way of doing a fish species inventory that doesn't involve going into the water up to your neck and hoping to catch the fish that are actually there in order to get an inventory. And for those of you who are at Windsor, this person in the front, that's actually Hugh McIsaac who's in the water. That doesn't happen anymore. Next, you have to be able to assess the health of the fish. Currently, if you want to determine whether a fish population is healthy, you grab a fish and you take it to a lab to do a necropsy. This is, first of all, time-consuming, it's lethal, and it's often inaccurate because necropsies are extremely difficult to do to determine what actually killed the fish. But it's even worse if all you're trying to do is figure out why is the fish not performing well, or is it performing well? And then the final key data gap is, what are the criteria for whether or not a fisheries manager needs to kick into the new tools or stick with what they've got? Maybe for Lake Erie yellow perch, they don't need anything new because they're doing a pretty good job, or maybe they're not. But the manager or the conservationist has to be able to say, how do I choose? Like, when do I make the jump? And spoiler alert, the solution is genomics. I've had a few people tell me that the genetics is a solution to everything as far as I'm concerned. It's the old adage that if all you've got is a hammer, everything looks like a nail. Exactly that. I'm a geneticist, so genomics is the solution. But wait, what are genomics? I suspect many of you know what genomics are, but maybe not the details. Well, genomics is part of a series of technology or approaches that have to deal with the expression of genes in individual cells or organisms. It consists of genomics that has to do with the DNA, transcriptomics that has to do with the messenger RNA, proteomics that has to deal with the proteins in the cell, and metabolomics, which is the metabolic byproducts of proteins being broken down. When you put these all together, the latest catch term is omics. But there's a problem, because for me anyways, I have this mental image whenever I hear the word omics, probably because I was dropped on my head when I was a baby. But whenever I hear somebody say omics, or for that matter I read, I think of a dog throwing up. This mental image distracts me, and consequently I have a hard time taking the term omics seriously. I use it when I have to because it's become a catchphrase, but for this talk anyways, because I'm in charge, I'm going to define genomics more broadly than simply the analysis of genomes. Rather, I'm going to say that genomics is the applications of genome-derived technologies. Well, what the heck does that mean? Well, the genome is the DNA in an organism, or actually in a cell. But the DNA is the basis of generating RNA through transcription. And DNA is ultimately the basis for producing protein through the process of transcription and then translation. So genomics, in my definition, is the study of any of the upstream or downstream components of gene expression that's derived from the genome. Simple as that. This way I get around having to say omics and keep thinking about that husky dog woofing his cookies. Okay, so now I'm talking about genomic solutions in fisheries. Okay, the first one that I talked about, the big question is, how do we know what's there? Are there fish there? What are they? Where are they? And this comes down to fundamentally fish surveillance. And for most of you probably heard environmental DNA, and I always like to put that picture over on the right, because if you Google images for eDNA, that's what you're going to get. And you have to be cognizant of Disney films to know what that's about. But anyways, the cool thing about environmental DNA is I'm going to be talking about fish surveillance. But there's an emerging group of individuals who are beginning to realize that, in fact, environmental DNA houses information that allows us surveillance of a lot more than fish. Everything from mammals to humans eating habits, etc. So the environmental DNA contains a tremendous amount of information that triggers all sorts of ethics concerns I'm not going to talk about, because we're just going to look at the fish DNA in environmental DNA. I'm going to give you a very basic primer on environmental DNA analysis. There are fundamentally two approaches. There are a lot of variations on these two approaches, but fundamentally two approaches. It starts with taking an environmental sample and extracting DNA from it. By far the most common environmental DNA source, at least in terms of what's being published, is actually water. So you take a water sample, you filter it, and you extract DNA from the crud that was in the water, and you can get a large amount of DNA extracted from that water sample or environmental sample. I'm going to be talking about water, but remember, people do environmental DNA in soil, they do it in air, they do it from leaf surfaces. So all sorts of there are all sorts of options for environmental sampling. Once you have the environmental DNA in your lab or in your hand, one approach is quantitative real-time PCR. And what this is PCR is DNA amplification, and quantitative real-time PCR is designed to pick up only one species And by its name, quantitative, it is quantitative. So it not only tells you that the animal or the plant or the organism is present, but it will give you an estimate of how strong the signal is. The limitation is it does one species at a time. This can be very problematic if you have a complex community and you want to know more than just is the species there or not. This leads to environmental DNA meta-barcoding. And what this is a very different application of EDNA analysis, where you use primers that don't amplify just one single species, but every species in the community. And then you use high throughput sequencing in order to determine which ones were there, because you're going to PCR amplify chunks of DNA from all these different species, ending up with a dog's breakfast of mixture of DNA fragments. But when you sequence it, you can use the DNA sequence to say, this species was here, and this species wasn't. Okay, the next proposed genomic solution in fisheries is fish health. If you've used environmental DNA to determine that a particular population is declining, or that there is evidence that they may start declining, the next question is, how do you determine whether or not this wild population of fish is healthy or not? In an actual wild population in a natural water body, this is complex, because if you capture a fish and do an assessment of health, it may not reflect the entire population. And as I mentioned earlier, assessing fish health is not easy. It's not easy with humans, and they can tell you where it hurts. Fish can't tell you that. So when you want to ask the question of, is a fish healthy, you can do transcriptional profiling. For example, if I were to take you and put you in a freezer overnight and pick you out the next morning and you're good and cold, you are thermally challenged and you're going hypothermic, I can look at you and tell you cold, but if I were to take some of your messenger RNA, I would be able to determine based on which genes are up and down regulated that A, you're not happy, and B, why you're not happy, that you were being thermally challenged, cold challenge in this case. We can do exactly the same with fish. It's a little more complex than that because there are problems with transcriptional profiling, but I'll be talking about that. So the issue behind stress response transcriptional profiling is that you have to look at more than one gene. Early in my career, I and my lab spent a lot of effort looking for the silver bullet gene. That is the gene where we could look at the transcription, and if it's upregulated, the fish are doing great. If it's downregulated, the fish is doing badly. So that silver bullet of fish performance. We failed miserably. And part of the reason, as we came to understand, is that the transcriptome, that is, the way that an organism expresses all of its gene, is not, oh, I'm going to respond to that by firing off this gene, or I'm going to respond to that by expressing that gene. Rather, it's an incredibly complex array of interacting gene functions that drives a response. So you see in this diagram, this is from humans, this is sort of a network diagram of how genes interact in different functions in a response in a human to normal everyday stresses. When you pile on top of the fact that nobody really cares about gene transcription or genes, and in fact, when you look at the genes that are associated with function in let's say, salmon, if you follow the publication ontology of the function of that growth hormone gene, it'll go back probably to zebrafish. And then, no, they didn't actually identify the function in zebrafish. It goes back to a mouse model where they actually did gene knockout studies. So we know what growth hormone does in mice, and we're assuming fish are the same. Growth hormone pretty safe, but a lot of the gene functions are probably wrong. So this means that the solution has to be using multiple genes that are in different parts of the gene expression pathway simultaneously. So if you've got one gene that doesn't actually function in the immune function of fish, It does in mice, but not fish. Now it's a stress response gene. It doesn't matter because you've got 10 or 20 stress response or immune function genes within this open array chip. The open array is the Thermo Fisher platform for doing this, and it can do 112 genes for 12 fish at a time. So you can look at gene transcription across an array of function, and that's going to give you an information that tells you how is the fish doing by relative up and down regulation of all those genes, that profile. But it will also say, look, all of them are fine except the immune function genes are going nuts. And they think, okay, there's some kind of pathogen in there that's bothering them. So it not only tells you that they're not well, but it gives you an idea, potentially, of why they're not doing well. Okay, I'm going to talk about applications. I recognize I left the criteria for adoption because this is a genomics talk, but I'm going to get to that at the end. Okay, so I'm going to give a couple of examples. They're just picked, sort of cherry-picked from work that my students have done. There's a lot more that have gone on in my lab, and if I went outside of my lab to some of my collaborators' labs, such as Ken Jeffrey or Nicholas Bernier, who I think are here, there's lots of cool stuff they've done, but I'm giving you this just as kind of examples. But before I do that, one of the things I learned early on in my profession is there are two types of people in the world. You can categorize them. That's something you're not supposed to do. It's sort of profiling. First type are normal people. That's most people. Second are geneticists. What I've learned is that when you're giving a talk, you have a choice. When I'm giving a talk, I have a choice. I pitch it to the geneticist and give all the details about what the annealing temperature is, how I designed the primers, how I know they're specific. And all that really functions to do is let the other geneticists know whether I'm an idiot or good at what I do. So it's not all that helpful even for the geneticists. Or what I do is I pitch normal people. And then the genomicists or geneticists amongst you, you can ask me questions about the details. So what I'm going to be doing is skipping over or glossing over all the really hard work by my students to make the results come out. And just take my word for it, it wasn't easy. Okay, so I'm going to talk about a project that was done by a master's student who was co-supervised by myself and Nick Mandrak. So that's Catherine Ballassingham. She's now Dr. Catherine Chow, but she started her illustrious academic career in my lab working on a project that was initiated by Fisheries and Oceans back when Nick Mandrak was with Fisheries and Oceans Canada. And the idea behind this was to use environmental DNA samples to assess the fish communities of Sydenham River and the Grand River, so two key river ecosystems in southwestern Ontario. And essentially, DFO sampled water at multiple sites up and down these rivers, shipped the water samples back to Windsor, and Catherine filtered them, extracted eDNA. The approach that Catherine took was to use this meta-barcoding. She's designed primers, PCR primers, to amplify fragment of DNA that should amplify from all Great Lakes fishes. So not only will she get her target species, so these were the species that were paying the bills, so to speak, but it would also get the rest of the fishes that might be in the water column nearby where the sampling took place. 3 of the species were species at risk: the silver shiner, the northern mad tom, and the eastern sand darter. 1 of them was invasive species round goby. So the real key reason why we got the money for doing this was for the species at risk, but of course there was concern the round gobies might be impacting these. So I'm going to skip all the hard work and just go to the results. Here we have a map of the Sydenham River. So there's Lake St. Clair, there's the north and eastern Sydenham River. What you see is each of the red dots on there, or each of the dots, is one of the sampling sites that DFO took a water sample and Catherine did eDNA analysis on. The red dot meant that she detected the round goby. A white dot meant the round goby was not there according to the eDNA analysis. And for technical reasons, there's one with a red with an X in it. That was failed quality control. We'd identified it as contaminated. It wasn't included. So you don't have to worry about that. So what Catherine's analysis showed pretty conclusively is round gobies have colonized the entire Sydenham River. Perhaps not a big surprise. The fact that they were right up at the top of the upper eastern Sydenham River was a bit of a surprise. The two sites that showed no round gobies were probably false negatives because we were being very aggressive to detect them. There you go. What about the Grand? Well, the Grand River showed pretty much the same result. This was a bit of a surprise to people involved because it was thought that the round gobies had not got across all the dams in the Grand River, but clearly they were introduced above the dams. You can see a number of sites in the upper Grand River that were free of round gobies, or at least free of round goby DNA. So there probably were at this time reaches of the upper Grand that didn't have the round gobies. My guess is by now round goby EDNA is throughout the river. What about the species at risk? Well, I'm not going to talk about all of them. I'm just going to pick one that's sort of interesting, and that's the northern mad tom. So this is the distribution of the northern mad tom in the Sydenham River. So we were able to detect this species at risk at 7 sites in the Sydenham River, including the north and the eastern Sydenham River tributaries. This was important because Department of Fisheries and Oceans knew that the northern mad tom had historically occurred in the Sydenham River, but they hadn't detected it with their traditional surveys in a number of years. And so this question of, do we identify it as extirpated and not stop worrying about its habitat? But when Catherine did this analysis, it's clear that there are extant round gobies in the Sydenham River, but they're hard to catch. They're a benthic fish, they're fairly small, they're fairly cryptic. So they're probably there, they're just hard to catch. They're likely in small numbers. Well, that was interesting, and Fisheries and Oceans were excited about that. But then there was the Grand River. There are no northern mad tom in the Grand River. Unfortunately, Catherine had a positive hit for the northern mad tom in the Grand River, in the upper Grand River. So at the time, Nick Mandrak and at that time his postdoc, Andrew Drake, were both very skeptical, with good reason, because they don't occur in this river. So anyways, Nick went on to become an academic, and Andrew Drake decided that we're going to redo this. We went and resampled, or Fisheries and Oceans resampled that region, as well as a couple of sites where there weren't any northern mad toms, just to keep us on our toes. And we reperformed this analysis, and we detected the northern mad tom DNA out of the same area a second time. And this begs the question of, are they really there, or is it some kind of artifact of environmental DNA? And at this point, I suspect Fisheries and Oceans still don't believe that the northern mad tom are there. And this highlights the acceptability or the criteria for adoption of eDNA technology in fisheries. Catherine also did a community analysis, because remember, her analysis metabarcoding is going to pick up all of the fish, not just her 4 target species. And community analysis is very important when you're dealing with species at risk, because you want to find out what type of communities of fish they prefer or they're found in. This is a coexistence plot. And what you see on the left side is the Grand River, on the right side is Sydenham River. And what you see are the fish that had some kind of non-random coexistence pattern. The light gray squares means random. That is the existence of Ictalurus punctulatus, and I don't know, let's pick up one of these, Natropus buchani, is random. That is, they don't particularly like to be together, nor do they particularly avoid each other. If in fact they were trying to avoid each other, you would see a white square. We found no evidence of avoidance. But what we did see, the dark squares, dark gray squares, are coexistence preference. So that's where they occurred together more often than you expect by chance. And what this type of data tells you is you take the common carp, which is a invasive species that is almost naturalized, and you can see that it tends to be found in places where other species are common. This is probably because carp like a particular habitat, and the other species like that habitat as well. Curiously enough, it's also the habitat that the round gobies tend to like. So the common carp and the round gobies tend to be found together more often than you would expect by chance, which was unexpected. So you can see the power of going beyond simply, I'm looking for round gobies and looking for communities. Okay, what about quantification? Can we quantify? So this project was by graduate student Alex Katar, shown here. And what he was interested in is, what is the EDNA plume or profile emanating from a commercial salmon farm? And so he sampled water at 3 different depths in a grid around the actual salmon farm in a bay in British Columbia. He sampled water, EDNA, at 3 depths over 3 days. He then used a Chinook salmon specific qPCR probe and primer to determine whether or not he could detect the Chinook salmon DNA. And he created like a contour map or a heat map showing where he could find the DNA and where he couldn't. And of course, what we expected was like a blazing hotspot at the net pens and then a gradual decline through dilution. That wasn't what was found at all. We found this chaotic pattern. So the light colored spots, like this one right here, that's high concentration of Chinook salmon DNA, and the dark blue are low concentration, and everything in between. So the interesting thing about this is, first of all, it changed day by day. So this is June 4th, 5th, and 6th, and you can see the pattern change dramatically. This is primarily due to sampling at different times of day, we think, and the tide was different. You can also see the depth made a huge difference. So what this is highlighting is, first of all, eDNA you can't assume is well mixed, and also that the way that the water's flowing is going to impact your ability to detect it. And you have to take that into account whenever you're using eDNA to say something is present or absent. So to summarize, eDNA is fairly widely accepted as a robust detection method, and it is at this point becoming commonplace for fisheries managers and conservationists to actually accept eDNA data. The community analysis is an exciting step forward or addition to just detection, and has some limitations, but it has strong fisheries values. And I think more and more people are realizing if you do a combination of the quantitative qPCR and the meta-barcoding, you have a really powerful data set to actually guide management decisions. The potential for this to become truly quantitative, where you can use eDNA to say how many fish were there, is still in the future. But preliminary assessment of this indicates that it should be possible. And some really exciting work's coming out of few labs in North America to move this from being presence absent to there were 10 fish there. I'm going to just quickly describe how transcriptional profiling can be used. And the example I'm going to be giving is work that was done by Shelby Toes, now Shelby Mackey, with her daughter Tessa shown there. So she did all the work behind this. And again, I challenge all you Windsor folks, or those of you from elsewhere who know Windsor, how many Windsor profs can you spot in that picture? The answer is 3. See if you know which ones they are. But anyways, the basic premise for Shelby's project was that a commercial fish farm, Yellow Island Aquaculture, wanted to improve the performance of their fish. And we looked into the potential of using transcriptional profiling. The basis of this project, which was multi-lab, there were 7 labs working on this, is we took gametes from 7 different stocks of Chinook salmon and crossed them with inbred fish at the fish farm, and then followed their performance, whether it be stress response, whether it be growth, whether it be behavioral characteristics, to determine which ones were performing best. But part of what Shelby was doing was really exciting, was to go beyond that and use these crosses not to determine which one's performing best, but which ones are going to respond to selection. That is, which ones could Yellow Island use as a basis and select for better performance. So what you see here is actually the outcome of that heritability analysis across the genes. So across the top, we have the genes that were part of the profile. They included immune function, growth, metabolic, and stress function genes. Because remember, we may think that IGF-1 is growth-related in fish, and probably is, but you want to look at multiple genes to get a profile, a pattern emerging. And remember, we're not looking at the level of expression or transcription of these genes, but this is heritability. This is telling you whether there is a scope for the population to respond to selection. And what you want is bright red. That indicates that a strong opportunity exists to select for really good immune function, really good growth or metabolic rate or stress response. And as you can see, the Puntledge and Robertson Creek crosses were the ones that on average had the highest heritability for these genes that are thought to be critical for performance in captivity. So that the outcome was, if you want to mount a selection program, this is what you want to use. Okay, what is the summary of this? Profiling over multiple genes has tremendous fisheries value because you can do it quickly. We're working on making it non-lethal, so you can take a non-lethal biopsy and get this information. And it's going to allow you to determine what genes are responding. In this case, it was sort of a different application to see which ones are heritable. But in normal applications, you just want to see whether the fish is up or down regulating across those genes. The problem is cost and throughput. That is, it costs a lot to do per fish, and it can take months or even years to get from having a fish sample to actually getting the data, which is not useful for fisheries. So along came GenFish. The purpose of GenFish is to use the expertise amongst our partners of 23 principal investigator labs scattered across Canada, as Tina mentioned, with a diversity of expertise ranging from eDNA to gene transcription to social sciences, to come together and come up with toolkits that are going to solve the logistic and technical problems associated with both eDNA and gene transcription in fisheries and conservation. Part of the power of GenFish is it's not just a bunch of us scientists getting together and talking to each other in omics language, but rather we have partners who are co-producing this with us. That is, we're interacting with them back and forth as we're developing these tools, and the potential is there. They can say, well, that's really cool, but it's totally useless. It's not going to address a problem for me. And then we are going to reposition and refocus, change our direction, say, okay, well, how do we make it useful for you? Because if we create these tools and they're not used, we just wasted time. So here we go. What we've got is genomics application in fisheries and fish conservation. eDNA is powerful. The STP stress transcriptional profiling chip is going to solve a lot of fish health assessment problems. Ta-da! Benefits. Well, as you might guess, there's a question mark there, and that is the GE⁻⁴ And for those of you who aren't in the know, this stands for genomics, ethical, environmental, economic, legal, and social aspects. So we were charged in GenFish to not just do the genomic science, but also address the social, economic, and other aspects of adoption of this tool. And until that is solved as well, and the three goals put together, we're not going to reap those benefits. And I'd like to acknowledge the agencies that supported the stuff I was talking about, and the individuals that actually did all the work. And I'll take questions if there's time.
Excellent, Daniel.
I'm going to stop sharing my screen. As long as everybody's had time to read my cartoon. There you go.